हेलो फ्रेंड्स, आज का टॉपिक है सेल द यूनिट ऑफ लाइफ पार्ट थर्ड। और आज ये चैप्टर कंप्लीट हो जाएगा। राइबोसोम, इंजन ऑफ सेल। Ribosome are the granular structures first observed under the electron microscope as dense particles by George Amplade 1953. They are composed of ribonucleic acid RNA and proteins and are not surrounded by any membranes. Except mammalian RBC, all living cells have ribosomes, both prokaryotes and eukaryotes. Ribosomes are smallest cell organelles. Ribosomes are also called as organelle within organelle and protein factory of cell. Types of ribosome. Eukaryotic ribosome 80S occur in cytoplasm of eukaryotic cells. Prokaryotic ribosome 70S occur in cytoplasm and associated with plasma membrane of prokaryotic cell. Their size is 15 to 20 nanometer. 70S ribosome also present in mitochondria and chloroplast of eukaryotes. S is equal to Swedberg unit or sedimentation rate. It indirectly is a measure of density and size. Each ribosome composed of two subunits, for example, larger and smaller subunit. 80S is equal to 60S plus 40S. 70S is equal to 50S plus 30S. Magnesium ion is essential for the binding the ribosome subunits. Magnesium form ionic bond with phosphate groups of rRNA of two subunits. Minimum 0.001 magnesium concentration is required for structural formation of ribosomes. Chemical composition of ribosomes. 70S 60% rRNA plus 40% protein. 80S 40% rRNA plus 60% protein. 60S rRNA 28S 5.8S 5S. 40S rRNA 80S 18S, 50S rRNA 23S 5S, 30S rRNA 16S. At the time of protein synthesis, several 70S ribosome become attached to mRNA with the help of a smaller subunit. This structure is called polyribosome or polysome or ergosome. Larger subunits 50S contains peptidyl transfer enzyme 23S rRNA which help in the formation of peptide bond during protein synthesis. This is an example of ribosome. Noller 1992. Cytoskeleton. An elaborate network of filamentous proteinous structure present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Microtubules. Microtubules are composed of protein tubulin size 25 nm. In plants, microtubules often found associated with cell wall. Probably these transport cell wall material from Golgi body to outside of cell. During cell division, these microtubules form spindle fibers. Microfilaments. They are composed of contractile protein actin which concern with muscles contraction. Microtubules and microfilament are part of cytoskeleton base of cell size 6 to 7 nm. Intermediate filament. Intermediate filament has size or diameter in between microfilaments and microtubules. These filaments form basket-like structure around the nucleus, size 8 to 12 nm. Cilia and flagella. Cilia single cilium and flagella single flagellum are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The bacteria prokaryotic cell also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscope study of a cilium or the flagellum show that they are covered with plasma membrane. Their core, called the axoneme, possesses a number of microtubules running parallel to the long axis. The axoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonal microtubules is referred to as the 9 plus 2 array, 9 doublet plus 2 singlet. Arms of a tubule consist of an enzymatic protein dynein similar to myosin of muscle cells. Dynein have ability of hydrolysis of ATP and liberates energy for ciliary or flagellar movement. The central tubules are connected by bridge and is also enclosed by a central sheath which is connected to one of the tubules of each peripheral doublets by radial spoke. Thus there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal bodies or blepharoblast. Diagrammatic representation of internal structure of cilia and flagella. Interdoublet bridge, peripheral microtubules, outer dynein arms, inner dynein arms. Bridge, central sheath, radial spoke. Cilia and flagella different. Cilia. The cilia are smaller in size, 5 to 10 micrometer. Number of cilia per cell is very large. Cilia beat in a coordinated manner, pendular movement. They take part in locomotion, attachment, feeding, and sensation. Flagella. First, flagella are long up to 150 micrometer, few in number. Flagella beats independently, undulating movement. Flagella involve only in locomotion. Centrosome and centrioles. Centrosome is absent in higher plants. Centrosome containing two centrioles. Diplosome located just outside the nucleus and lie at right angle 90 degree to each other. Each centriole is surrounded by amorphous pericentriolar material. Centrioles are membraneless cylindrical structure which exhibit cartwheel structure in transverse section. Centriole mainly consist of 9 evenly spaced peripheral triplet fibrils of tubule. In these triplets are linked with the help of AC linker. The central part of the centriole is proteinaceous and called the hub, which is connected with peripheral triplets by radial spokes made of protein 9 plus 0 arrangement. Centrioles are self-duplicating units. Centrioles. Triplet microtubules. Muscles or pericentriolar satellite. AC linker. Triplet fibril. Centriole hub. 9 radial spoke. Function. In animal cells, centrioles play important role in cell division by arranging spindle fibers between two poles of cell. The location of centrioles during cell division decides the plane of division. The plane of division is always at right angle to the spindle. Centrioles form the basal body of cilia or flagella. Microbodies. These are many membrane-bound minute vesicles contain various enzymes that are present in both plant and animal cells. Number first, peroxisomes or uricosomes. These are found in both plant and animal cells. Peroxisomes contain catalase enzyme which is concerned with peroxide H2O2 metabolism. Catalase degrade the H2O2 into water and oxygen. In plants, peroxisome occurs in cells of green tissue and concerned with photorespiration glycolate pathway. Peroxisomes are also involved in beta oxidation of fatty acid. Glycosomes. Glycosomes occurs only in plants, especially in fatty acid, castor seed, groundnut seed, etc. Glycosomes are considered as a highly specialized peroxisome. Glycolate acid cycle takes place in glycosomes. This cycle converts fat into carbohydrates. Nucleus. Introduction. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus sustained by the basic dyes acetocarmine was given by name chromatin by Fleming. Nucleus is double membrane bound dense protoplasmic body which controls all cellular metabolism and includes the genetic information of cell. Nucleus is considered as controller or director of cell. Importance of nucleus is control of heredity. Growth and metabolism was experimentally proved by Hemmerling. Experiment was on Acetobularia, a single cell largest alga. Generally, eukaryotic cells contain at least one nucleus, but nucleus is absent in mature phloem, sieve tube elements, and mature RBCs of mammals. Dikaryotic, paramecium, and multikaryotic cells are also known as. A structure of interphase nucleus. Interphase nucleus of cell when it is not dividing. Number first, nuclear membrane और कैरियोथिका. नंबर सेकेंड न्यूक्लियर मैट्रिक्स या न्यूक्लियोप्लाज्म या कैरियोलिम्फ या कैरियोप्लाज्म. नंबर थर्ड क्रोमेटिन नेट. नंबर फोर न्यूक्लियस या लिटिल न्यूक्लियस या राइबोसोम फैक्ट्री. रफ एंडोप्लाज्मिक रेटिकुलम न्यूक्लियर पोर्स. न्यूक्लियर मेम्ब्रेन. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nm, called the perinuclear space. These membranes form a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains contains with the endoplasmic reticulum and also various ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores which are formed by the fusion of its two membranes. These nuclear pores are the passage through which movement of RNA and protein molecules take place in both directions between the nucleus and the cytoplasm. The nuclear pores is guarded by a octagonal discoid structure of nucleoplasmin protein. The inner side of inner nuclear membrane is lined by nuclear lamina. This structure is formed by filament of laminin protein, nucleoplasm or karyolymph. Nucleoplasm or nuclear shape is a ground substance of nucleus which is a complex colloidal form of a number of chemicals like nucleotides, nucleosides, ATPs, protein and enzymes. Chromatin, net and nucleus are components of nucleoplasm. Chromatin net, term given by Fleming. Interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin which embedded in nucleoplasm. Chromatin net is mainly formed of DNA and histone protein complexes. Chromatin fibers contain genetic information and condensed to form chromosomes during cell division. During different stages of cell division, cells was structure chromosome in place of nucleus. Chemically, chromatin consists of DNA, RNA, histone protein, basic proteins rich in arginine and lysine, and non-histone protein. Chromatin net has two types of chromatins. Euchromatin. This is lightly stained and diffuse part of chromatin which is transcriptionally or genetically more active. Heterochromatin. This is dark stained, thick and condensed part of chromatin. Heterochromatin is genetically less active or inactive chromatin. Constitutive heterochromatin occurs in all cells in cell stage, for example, centromeric region. Facultative heterochromatin occurs in some cells in some stage, for example, par body in females. Nucleolus. The nuclei are spherical and membraneless structures so that the content of nucleus is continuous with the rest of the nucleoplasm. It is a site for active ribosomal rRNA synthesis. Nucleolus usually attached to chromatin or chromosomes at a specific site called nuclear or organizer region (NOR). Nucleus is called ribosome factory of cells. Larger and more numerous nuclei are present in cell actively carrying out protein synthesis. Chromosomes. General introduction. At the time of cell division, the chromatin material gets condensed to form chromosomes. Thus, chromosome is a highly condensed form of the chromatin. Chromosomes are not visible during interphase stage, but during different stage of cell division, cells show a structured chromosome in place of the nucleus. Chromosome can be best studied at metaphase stage because size of chromosome is the sorted during metaphase. Shape of chromosome is studied at anaphase stage. Chromosome numbers in myocytes diploid 2n and gametes haploid n of same organisms. Chart of chromosome numbers in animal. The number of chromosomes in a gamete is called genome, or a complete set and of chromosomes inherited as a unit from one parent is known as genome. A single human cell has approximately 2 meter long thread of DNA distributed among its 23 pairs of chromosomes. Types of chromosomes on the basis of position of centromere. Telocentric, when centromere is terminal or located at the tip of chromosome. Acrocentric, when the centromere is subterminal or located near the tip. Metacentric, when the centromere is located at mid of the chromosome. Submetacentric, when the centromere located near center or midpoint of chromosome. The ratio of length of the long arm to short arm of a chromosome is called arm ratio. Arm ratio is maximum in acrocentric chromosome. Telocentric chromosome, acrocentric chromosome, metacentric chromosome, submetacentric chromosome. Karyotype. Karyotype is external morphology of cell chromosome of a cell which is specific for each species of living organisms. Karyotype can be studied in metaphase of mitosis. Karyotype includes the number of chromosomes, relative size, position of centromere, length of the arm, secondary constructions, and binding patterns. Idenogram. Diagrammatic representation of karyotype. In ideogram, chromosomes are arranged in decreasing order of size. Sex chromosomes are placed in last. Ideogram is specific for every species. Structure of chromosome. Number first, chromatid. At metaphase stage, each chromosome is consists of two cylindrical structures called chromatids. Both sister chromatids are joined together by a common centromere. A chromosome may have single chromatid in anaphase or telophase, or two chromatids as in prophase and metaphase. Each chromatid is consists of a single long thread and DNA associated with histone, non-histone protein and RNA are also present. Karyotype. Karyotype is external morphology of a अच्छा ये हो चुका है. Centromere. Each chromosome at prophase or metaphase is consist of two chromatids. Both the chromatid of a chromosome are joined or connected by a structure called centromere. At centromere. Two proteins discs are present which are called kinetochores. Kinetochores constitute the actual site of attachment of a spindle to chromosome during cell division. At the region of centromere, the chromosome is comparatively narrow than the remaining part of chromosome, thus it is termed as primary construction. Figure of chromosome with kinetochore. Kinetochore. Secondary constructions. Besides primary constructions, other constructions may also occur on some chromosomes which are known as secondary constructions. These constructions are non-stunning and found at a constant location. Secondary construction is also known as nuclear organizer region 13, 14, 15, 21, 22 chromosome in human. Kinetochore. Primary constriction or centromere. Secondary contraction. Telomere, satellite. Telomere. Satellite. Part of chromosome remain after the node is known as chromosome satellite. Telomere. Chromosomes have a polarity and polar ends of chromosome are known as telomere. Telomere prevents fusion of one chromosome to other chromosome. Telomeres are rich in guanine base 5 TTAGGG3. Enzyme telomere synthesize telomere part of chromosome which is a ribonucleoprotein. Telomere of chromosomes become shorter during aging process. A special type of chromosome. Number first, salivary gland chromosome. This type of chromosome was discovered by E.G. Balpiani in Chironomus larva. This chromosome is called polytene chromosome because number of chromatids are very high. Swollen areas present at some places in polytene chromosome which are called as Balpiani rings or puffs. These puffs help in synthesis of RNA and protein. Salivary gland chromosomes concerns with metamorphosis and molting process of insect larvae. Lamprey chromosome discovered by Fleming and Ruckert from oocytes of vertebrates amphibia during diplotin stage of cell division. These chromosomes look like lamp brush, thus called as lamp brush chromosome. Axis of lamp brush chromosome is consist of DNA while matrix is consist of RNA and proteins. Lamp brush chromosome is concerned with vitellogenesis yolk formation. Filament DNA. Chromosome axis. Matrix protein plus DNA. Chromosome pair.